answer. Okay, meiosis refer to nuclear division. It's a type of karyokinesis. But why we have a new term here? Why we can't use mitosis anymore? Because meiosis is a process that the nucleus will divide twice. Kita akan membahagi sebanyak 2 kali. Okay, sebelum berlakunya meiosis, we have interface. Okay, they still have interface. Same goes to the cell cycle we learned before. Okay, so the interface, they have 3 surfaces that we learned before also. Okay, in this order. Okay, which one is the longest subphase in interface? Okay, the first gap phase is the longest phase. Okay, in the interface. Okay, the chromosomes still in chromatin form. Okay, from the G1, the first gap until the second gap. Okay, we always start with the diploid cell because, as I say, when we study a normal cell, okay, diploid organisms, the body cells are diploid cells. Means that each type of chromosome in the nucleus, we found 2 copies, or we say 2 sets of chromosome in that nucleus. How we count the 4 chromosome? We always look at the centromere. But unfortunately, as I say, this is chromatin, so somehow difficult for us to identify the centromere. So I just need you all to recall what we have learned in theory class. In first gap, we know that all chromosomes, they are in the condition we say unduplicated chromosome. Okay, unduplicated chromosome. That means the chromosome, there has only one chromatic. Okay, so this is the picture we know. So here we can't see the centromere, but we can try to understand each fiber-like structure. Setiap structure yang macam berbenang halus ni mewakili satu chromosome. The chromosome number in S phase still 4. Okay, still 4. But you look at these 2 pictures, okay, the nuclear content in the S phase seem like clouded. Dia macam tiba-tiba jadi meriah di sini. DNA replication happen means now our chromosome is called as duplicated chromosome because each chromosome, their DNA already replicated to produce another identical DNA molecule. So now the chromosome is called as duplicated chromosomes. Okay, so can you suggest me the DNA amount in the nucleus of S phase? Kalau kita ada DNA replication, yes, that's why the amounts of DNA will be double. Good. Okay. And we move on to the last gap, the last subphase in interphase. Okay. In the last subphase, the chromosome number and the DNA amount will be seen from the previous. Why? Now we already complete the S phase. And the second gap, as we learned before in the theory class, second gap is actually a subphase prepare for the next activity, which is the cell division. So today we are going to study the cell division, which is meiosis, bukan mitosis. The centrosome duplication happens. So you have 2 centrosomes. Do you notice? Okay, and remember here, each centrosome, we have a pair of centromes. So we have to be careful with the keyword centros and centrosomes. Meiosis 1 consists of 4 phases, ada 4 phases. Okay, can you tell me which one is the longest one? Meiosis 1 until meiosis 2, prophase 1 is the longest stage. Okay, but you can compare the drawing. Kalau saya kecilkan, you can compare the drawing of the chromosome, right? Prophase 1, we can see the changes of the chromosome shape. They appear like the chromosome we always understand from the book. It seems like a butterfly already, because we want the student to understand that here is the happening of the condensation. The chromatin start to shorten and thicken, become the visible chromosome under the microscope. But I think you're able to tell me here how many chromosome number now in prophase 1?
4.
In one cell, yes, there are 4 chromosomes. Okay, now you can see the centromere very clearly because condensation happen. Tadi dalam interface, condensation tidak berlaku. So maka susahlah untuk kita cari centromere. But you can notice that the chromosome number and the DNA amount should be continued from the second gate. Jadi chromosome number masih 4 and the DNA will be 8. Okay, sebab kita akan nampak 8 DNA. Okay, setiap kromosom ada 2 sister kromatik. Kalau 4 kromosom, so 8 DNA. So how about the metaphase one?
Masih 4.
Okay, masih sama, masih 4. DNA amount pun masih sama. Okay, tak ada perubahan sebab my keyword is per cell. But the metaphase one, be careful, is when we talk about their chromosomal behavior, right? Okay, we have to call this chromosome in a proper term. Okay, what is aligned at the metaphase plate now?
Homologous chromosome.
Betul sekali, ya. So here we have 2 pairs of homologous chromosomes, a bigger pair and a smaller pair. So you will notice that each pair, they always represented with different color to represent different source of the chromosome. So if you notice in Campbell, somehow they call this as homolog. Okay, they use the term homolog. Homolog means the member chromosome. Okay, the member chromosome. So homologous pair got 2 homologs. Okay, and compare with the mitosis we learned previously, the metaphase in mitosis is 1 centromere attached to 2 microtubules from opposite poles of the cell. But in metaphase 1 centromere, they will attach to 1 spindle fibers only. Because why? The centromere of another homolog, okay, will attach with the microtubules from opposite pole. Okay, so this is the main difference here. We can see the metaphase of mitosis and the metaphase 1. Okay, how about we look at the anaphase 1 class? Is there any changes to the chromosome number and the DNA amount? That's why di sini sampai sini kita punya DNA amount constant. Okay, kita kata dia tidak mengalami perubahan. Sebab walaupun dia menunjukkan perpisahan, tetapi in one cell, we still notice 4 chromosome, 8 DNA. That's why in lecture note, we highlight here this activity. What is separating and moving towards opposite poles?
Homologous chromosome.
Betul, Fiza. This one is the separated homologous chromosome, which is totally different with the anaphase in mitosis. We notice that the separate and the moving one is sister chromatics. So you can notice that now the separated chromosome, they still carry their 2 sister chromatics. Dia masih membawa 2 sister chromatics dia. Okay, we move on to the last one, telophase 1. Okay, telophase 1 means that this is the end of the nuclear division. Lengkaplah, okay, nukleus itu menunjukkan pembahagian yang pertama. Sebab kamu dapati bahawa the nuclear envelopes reform. Okay, bentuk semula. This is why they always with a characteristic of the cell shape. Bentuk sel tu secara teorinya kita menunjukkan ada cleavage furrow. Okay, to show how the cytokinesis follow immediately. Dia bermula terus dengan cytokinesis. But you be careful class. Okay, now my table here still refer to 1 cell. Okay, masih kita tengok cell ya. That's why you look at the chromosome activity in 1 cell. Maksudnya kita apa-apa yang berlaku dalam membrane ini. Bukan tengok nucleus dia. Okay, can you tell me how many chromosome in this cell? So you have to focus on the activity happen surrounded by 1 membrane, still 1 cell membrane. So I still have 4 chromosome in this membrane class. Okay, how about the DNA amount?
8.
Yeah, 8. Sorry, I can't. I think my this one got problem. Yes, 8 DNA. Okay, but just now when you all tell me 2, this one is referred to this condition. The nucleus. The cell in telophase 1 here, they already form 2 nuclei. This one menunjukkan 2 nuclei. And now these 2 nuclei different with the daughter cell in mitosis. These 2 nuclei we call it as haploid nuclei. Okay, why is haploid nuclei? Because you notice that each chromosome is only found 1 copy. satu salinan sahaja, iaitu kromosom yang bersaiz besar ini, dia satu salinan sahaja. Kamu tengok tadi Prophase One dia ada dua. Okay, satu besar, kedua yang besar. Tetapi yang besar ni sekarang tinggal satu dalam nukleus ini. Yang kecil pun tinggal satu. That's why kita panggil dia haploid. Tetapi kalau soalan kita tanya the chromosome amount in this nucleus, then barulah kita kata in the nucleus we have 2 chromosome. How many DNA class? Ya, 4 DNA. DNA kita kira dia punya chromatic. Okay, 4 DNA. So, as I said, telophase 1 follow immediately by cytokinesis. So, tengah serta-merta berlakulah pembahagian cytoplasm. Okay, this is why we expect student to understand when the cytokinesis completed, they are 2 daughter cells are formed. And each daughter cell are haploid daughter cells because they have the nucleus which is haploid. So I continue with the amount that you suggest just now, where we know that in the nucleus they have 2 chromosomes and 4 DNA. And now they continue, okay, the second times of nuclear division, which is the meiosis 2. Okay, meiosis ya, still the nuclear division. Tapi ini kali kedua already. But in between the meiosis 1 and meiosis 2, we have a phase. Kita ada satu fasa ya. Tetapi fasa ini kita tak boleh panggil dia interphase anymore. Because they are different with interphase. They do not have S phase. They do not have S phase. That's why we introduce a new term. We call it as interkinesis. Kinesis means division. Hanya nak menunjukkan ada satu fasa dijumpai antara dua nuclear division. That's why nama dia interkinesis. Okay, so one more time. If we say that this phase do not have the S phase, so means no DNA replication. Jadi DNA amount kita akan kekal ya bila dia proceed to prophase 2, the first phase in the meiosis 2. Okay, itulah kenapa kita punya DNA amount itu nampak tak dalam total cell ini yang dia telah menunjukkan kromosom contents ini. Kromosom dia masih 2 kromosom, DNA dia masih 4. Kita tidak mengalami double. Okay, atau kita kata replication itu. Okay, dah seperti tadi, sebelum dia masuk ke meiosis 1, dia mula dengan parent cell yang 4 DNA. Lepas S phase dia dapat 8 DNA. Okay, dengan 8 DNA mulalah okay, journey dia untuk meiosis 1. Tetapi bila dia nak masuk ke meiosis 2, DNA itu tidak mengalami replication. This is why we continue with 4 DNA, 2 chromosomes. Okay, so in metaphase 2, you notice that, one more thing class, do you notice that, can you compare this cell, okay, kalau saya kecilkan dia, okay, can you compare, okay, the cell that start the prophase 2 and the daughter cell after the meiosis 1, right? Can you focus on the centrosome? What happened to the centrosome, class? You notice that after the interkinesis, yeah, after the interkinesis, we have 2 centrosome back in the cytoplasm. Tadi semasa okay, di hujung telophase 1, sebenarnya setiap daughter cell akan dapat 1 centrosome sahaja. Ini adalah sebab interkinesis, even though they do not have the S phase, but they still have the first gap and the second gap. Okay, that's why if they have the second gap, the centrosome will be duplicated. Jadi datang semulalah 2 centrosome itu. Okay, so kita move on to metaphase 2. So metaphase 2, each cell how many chromosome? Kita tengok satu cell ni, still 2 chromosome, and then the DNA amount still same. Okay, and now this is why I keep telling students understand the meaning of Campbell. The meiosis 2 is similar with the mitosis because you notice now the chromosome, the centromere, will attach to the microtubules from opposite poles of the cell. Okay, so same centromere attached to the microtubules from opposite poles of the cell. Okay, just the difference is compared with mitosis, now in this cell we have only haploid set of chromosome. Sebab saya tak nampak ya kromosom yang sama jenis dijumpai. Ya, mereka satu set sahaja. Jadi teruslah kepada anaphase 2. Anaphase 2, okay, we focus one cell here. Of course, we know that there are two daughter cells will perform the same thing. This is the first daughter cell, and we have the second daughter cell. But if I focus on one cell, how many chromosomes now? Yeah, we have 4 chromosomes. Okay, 1, 2, 3, 4. Look at the centromere. Okay, look at the centromere. Same goes to another daughter cell here. 1, 2, 3, 4. Okay, why we have 4 chromosomes? Because now the sister chromatics, ini sister chromatic yang tadi kita faham. Ya, that's why saya mula dengan terminologies tu. They have 2 sister chromatic, okay, in the metaphase. In the metaphase 2, each chromosome have 2 sister chromatics. But when they move on to the anaphase 2, sister chromatics separate and move to opposite poles of the cell. When sister chromatic separate and move, right, they will become the chromosomes. That's why we can use the term, how many chromosomes in the cell. So there are actually 4 chromosomes. But the DNA amount, how many? Yeah, Lija, okay, good. There are 4 DNA. So you see, now the DNA, you notice they are constant. Okay, we move on to the telophase 2. Okay, telophase 2, they will perform the similar thing here. Each daughter cell, setiap daughter cell yang dibentuk daripada meiosis 1 tu, bila dia lepas meiosis 2, right, they will develop nuclei in their cytoplasm. Dia akan membentuk 2 nuclei. Okay, of course, each nuclei we still call it as haploid nucleus. Dia masih haploid sebab kromosom tu hanya satu set sahaja. Okay, satu set sahaja. Okay, only when the telophase 2 followed immediately by cytokinesis. As I say, telophase 2 symbolize the end of nuclear division. Dia mewakili tamatlah, okay, pembahagian nukleus itu. Jadi tengah serta-merta mulalah cytokinesis. Once cytokinesis complete, now each daughter cell will form 2 daughter cells. But as I say, because we start with 2 daughter cell from meiosis 1, disebabkan kita punya meiosis 2 bermula dengan 2 daughter cell, maka kita dapatlah 4 daughter cell after meiosis 2. And this daughter cell, they have a special term. We call it as gamete. Because in biology, only gametes, they are haploid cell. Okay, hanya gamete saja dia dipanggil haploid cell. Sel-sel yang lain adalah diploid. Okay, so from our case, right, this haploid cell contains how many chromosomes?
2.
Yes, each daughter cell, they only carry 2 chromosomes. So, kamu imagine bila cytokinesis sudah complete, maka kita dapat 1 kromosom, 2 kromosom. And how many DNA in the nucleus of this daughter cell?
2 juga.
2 juga. Okay, 2 juga. Okay, so now you can see the DNA amount ya, class. Kamu boleh tengok DNA kita. Okay, perubahan DNA. Okay, the amount of DNA, they show changes. Okay, they show changes in parent cell. Okay, I use this example only. In parent cell, I start with a parent cell, okay, which is 4, right? Okay, after S phase is become 8. So I start the parent cell, which is 4. After S phase is become 8 DNA. And you see, after the meiosis 1, okay, after the meiosis 1, each daughter cell will carry 8, still 8, yeah? Still 8 DNA. That's why they're constant di sini. But after meiosis 2, how many DNA?
4.
We have after meiosis 2 in each cell.
Oh,
Each cell. Oh, saya salah tulis. This one should be 4. Okay, sebab kita kena tengok each cell, ya? Each cell. Okay, 4. And after meiosis 2, how many?
2.
2. That's why you can see DNA amount will be half twice. Sebab nuclear division happen twice. That's why the DNA amounts will be half twice. Akan diseparuhkan sebanyak 2 kali. Okay, tujuan dia separuhkan kepada 2 kali untuk memastikan later on when the gamete fertilize with the gamete, they will produce a normal diploid zygote. Okay, so this is how they maintain the chromosome number and DNA amount in a species. Kita tak mungkin dapat new generation dengan DNA amount yang tiba-tiba berkali ganda atau berkurangan. Okay, so when the zygote is formed, when this type of organism is formed, of course they will continue to their own cell cycle to become multicellular organism supaya dia boleh menambahkan bilangan sel tu menjadi satu organism yang normal. Okay, so this is the cell cycle we study in the first subtopic. Okay, how from one diploid zygote cell, they able to complete the complete cell cycle, which is the mitotic phase happen here. Maksudnya dia melibatkan mitosis daripada satu sel dia dapat two total cells here, and 2 get 4, 4 get 8 and so on. Okay, so kelas sampai sini boleh faham? Sampai sini boleh Okay, sampai sini boleh faham? Ya, kalau kita habiskan sikit lagi, can you suggest me how about the changes of chromosome number? Kita guna contoh ini untuk senang faham. So chromosome number, I start with the parent cell. This is 4. Okay, I start with 4. Okay, and I notice after S phase, the chromosome number still same because they just double the DNA. Okay, they only double the DNA, but the chromosome number still same. After the meiosis one, right, I noticed that the chromosome number has been half once, telah dibahagikan sekali. Tetapi after meiosis 2, after meiosis 2, I noticed that the chromosome number remain unchanged. Kamu tengok chromosome number dia kekal. Kekal. Ini yang kita selalu kata meiosis 2 dia mematuhi konsep mitosis. Because mitosis say that two total cells are formed from one parent cell, and the total cell carried identical chromosome number from each other and with the parent cell. Okay, itu yang kita kata kalau kita study meiosis tu kan, saya mula dengan sel yang 2 chromosome number. So selepas lengkap meiosis tu, the total cells still have 2 chromosomes. Okay, so konsepnya ialah DNA amount dibahagi sebanyak 2 kali, tetapi chromosome number hanya dibahagikan sekali sahaja pada meiosis 1. Okay, untuk menghasilkan haploid total cell.